change if
no, we don't mind.
Any strong views.
Okay, so we're live on YouTube. Are we all set with pre-presentation prep? Everybody good?
I think so, yeah.
Okay, great. So we'll just get started in about, I'd say like 2 minutes. Give people time to join up.
There's always so many people that register like right now to send all the emails out.
Uh-oh. Yeah, I mean, we're not one of those operations that has like all the fancy automated email sending set up.
We need to get that set up.
How? If I can help, Adam, let me know.
That's all right. I'm used to it now. Okay, I think we can get started. I've done all my admin stuff. Okay, so hi everyone. Thanks for joining us again. So this is our third from last seminar before Christmas. And we have 2 speakers today. So same as usual, so if you're new, you can pop your questions in the chat, which I can read out to the speakers afterwards. Or if you'd like to ask the question yourself, you can just write the word question and I can ask you to unmute afterwards so you can ask it yourself. Okay, so our first speaker today is Erik Sahai. Erik is the head of the Tumour Cell Biology Lab at the Francis Crick Institute in London. His research focuses on the spread of cancer through the body and responses to cancer therapy. In particular, his group is interested in stromal fibroblasts and their interplay with tumours and immune cells using a wide range of techniques from computational modelling of cell migration through conventional cellular molecular biology to intravital imaging of mouse tumours and live analysis of patient-derived material. Erik is has been a recipient of the Hook Medal, elected to EMBO and the European Academy of Cancer Sciences, and he's also the president-elect of the Metastasis Research Society and serves on the editorial boards of several journals, including JCB, DevCell, and Trends in Cell Biology. So Erik, thanks a lot and take it away.
Well, thank you, Adam, for that kind introduction. I will endeavour to share my screen and then get cracking. So hopefully you can all see a title slide now. So thanks, Adam and the other organisers for inviting me to present and be a virtual speaker in this great series. So I will kind of focus on the first sort of topic that you can see here, which is the determinants of pattern in collective cancer cell invasion. And then we will hear from Hamid, who will tell us about the role of matrix plasticity in cancer cell and CAF communication. So I'll just start with a little sort of introduction into the problem that we're interested in. So this is a histological section of an invasive squamous cell carcinoma. Just to give you a bit of scale, it'll be about 1 centimetre across. And hopefully I can indicate on the screen that it would have started somewhere kind of up here, as it were, and it's invading down in this direction. Just to kind of highlight what we're kind of interested in, these sort of slightly darker purple things that I'm outlining are the kind of collectively invading cancer cells that you can see here, and they're invading down in this direction into the underlying dermis in this case. And it's not just about the cancer cells, there are many other components to this system. Of particular note are the cancer-associated fibroblasts, and now what you're looking at is a serial section of that same tumour, but stained in brown are the cancer-associated fibroblasts, and we'll hear a lot more about those later. And you can see that they, in some cases, organise the extracellular matrix to be almost like a barrier, kind of at the edges of this tumor, and in other cases, they make the matrix highly aligned in parallel with the collectively invading cancer cells. The third kind of component that's really important for today's talk is actually kind of between the cancer-associated fibroblasts and the cancer cells here, and it's this stuff that isn't actually stained, but is the extracellular matrix, and we'll hear more about that in the second part of the presentation from Hamid. So if we just kind of think about how this system might work over the past decade or so, actually we've done really kind of quite a number of studies trying to document how cancer-associated fibroblasts drive this kind of invasion of the cancer cells. And this has told us a number of things. One is that cancer-associated fibroblasts are often at the front of these kind of invading strands of cancer cells that you can see behind. The fibroblasts play an important role in remodeling the extracellular matrix, and actually Cedric Gajoli, when he was in the lab, was able to show that one of the things that they do is they kind of generate these permissive tracts or tunnels that you can see that cancer cells are then able to exploit. More recently, we've been working together with Javi Trepat to understand this interface that sometimes occurs between the cancer-associated fibroblasts and the cancer cells that follow behind them. And really, through working with Javi's group, we were able to show that there's mechanical coupling in some cases between these two cell types, meaning that the fibroblasts really are pulling the cancer cells behind them. But what I'm really going to kind of concentrate on for the next 20 minutes or so is actually this kind of aspect of the pattern that's kind of emerging behind the cancer-associated fibroblasts, and that is how those cancer cells are, organizing themselves. And our interest in this kind of really, came from, again, staring at many of these, histological sections of tumors. And again, this is another one of these squamous cell carcinomas that would have started somewhere at the top up here. But what I hope you can appreciate is that actually the cancer cells in the kind of darker pink color are organized in a range of kind of different ways in these different boxes. Here you can see that they are sort of in these rather roundish kind of broad sort of clusters with many neighbors. Here they're in these kind of slightly thinner kind of groups here. And actually here it almost looks like they're kind of single file as I'm outlining. And we can quantify this just kind of by scoring the number of neighbors that a particular cancer cell has. And this is plotted on the right-hand side as you're looking at it. And it kind of starts to paint a picture that there are these sort of, wide range of patterns that cancer cells in these types of squamous cell carcinoma can use to invade. Probably a continuum between the very broad to the very thin strands that are observed. And if we, don't just look at fixed sections, but actually we get some of the patient material, again, from, the surgeons and our clinical colleagues. And in this case, we just kind of embed it within a three-dimensional extracellular matrix. We can then make movies of these cells invading. And what you can see in kind of this top example is that, we've got some cancer cells moving out from the bulk of the tumor material, which would be here. And you can see them kind of invading off into the surrounding matrix. Some of them have nice lamellipodia. There's a nice sort of example here of a cell that starts off having some blebs, and then it actually transitions to switching with a more kind of, actin protrusion-driven kind of mode of migration. But for today's talk, we're actually interested in these other three movies that I'll just start to play. And what I hope you can appreciate in these movies is that, again, we see this diversity of behavior from a single cell sort of, single file migration that you can see here that actually splits into cells, individual cells at the end, to these kind of, slightly thicker collectives that you can see here, to a very broad sort of pushing front that you can see in the bottom right movie. So this really, I think, sets the scene for what we would want to understand, and that is, what is it that, determines whether the cancer cells are behaving, like this, or whether they're behaving as you can see in this bottom panel here. And the fact that within the same tumor we can see, all these kind of, different behaviors tells us that it's probably not something connected to the sort of hardwired genetic drivers of this tumor, but actually it might have, more sort of, different origins. So what we sort of, set out to do a few years ago now was to really, understand what determines the collective organization of these invading cancer cells. I guess when we were thinking about this problem, we had, a range of possibilities in mind. It could be, cell-cell adhesion, cell matrix adhesion, the extracellular matrix and its proteolysis, or maybe, other cues. And I'm sure you may have, your own ideas about what might be the differences between the types of invasion pattern that we observe. And our approach to this was actually to span both computational approaches and an experimental model. And the reason for kind of engaging with computational modeling is that with all these different sort of parameters in mind, actually trying to understand how they might intersect gets very challenging if one goes at it just using an experimental system. So actually having a computational model where you effectively have perfect control and isolation of your variables seemed like a very fruitful way forward. So just a little bit of background about the POTS modeling, and I should really acknowledge that this has been led by Rob Jenkins in the lab. So for those of you who aren't familiar with POTS modeling, it's a type of agent-based model, and each cell is kind of composed of several sort of hundred voxels. So this is just a kind of 2D representation of the cell, kind of shown in green here. Looks a little bit like a sort of 80s video game, or for a more up-to-date reference, a bit like a sort of Minecraft world or something. But the bottom line is that each cell has all these different voxels, and then at each time step in the model, what can happen is that the identity of one of those voxels, or those voxels, can change. And what determines whether the identity of a voxel changes is really the various costs associated with transitions between that voxel state. So they have different sort of costs associated with them. So they're the kind of key input parameters, and depending on calculating the sort of cost or energy minimization, the model then updates the position of or the identity of that voxel. And if a contiguous group of voxels gets updated in a slightly adjacent position, that kind of gets recorded as cell movement. So the sorts of things that we can play around with are cell size. We can put in directional cues, cell adhesion or cell matrix adhesion, and also we can have the cells endowed with an ability to degrade or push the surrounding extracellular matrix. So those are the sort of principles of the model. Just to kind of take you through a little bit of simple validation, we ran a couple of mock experiments, either in the context of a spheroid, and I'm sure I don't need to introduce spheroids, it's just a ball of cells embedded in a three-dimensional extracellular matrix, or because it's a skin cancer that starts off as a kind of layered epithelium with underlying extracellular matrix and fibroblasts. We also have this kind of context of assay, which we call organotypic, where we have a layer of cells that is sitting on top of some extracellular matrix, and actually the model predicts if there are no fibroblasts present, you get no invasion, and this is borne out in the experiment. But if you have fibroblasts, these sort of magenta dots that you can see here present, then the model predicts that the cancer-associated fibroblasts will invade down into this kind of surrounding dermis, or they will invade radially if we have the spheroid kind of context. And of course, this isn't really news. This is just some validation that the model was able to recreate some of the behaviors that we'd already observed. But with this model in mind, we could then sort of play around to our heart's content with the various different sort of parameters that we hypothesize might be determinants of the pattern of collective cancer cell invasion. And this is really going to be what I'll tell you about in the next sort of 10 or 15 minutes. So the first thing was that just by playing around with these parameters, we could get various different sorts of invasive patterns. So if you look here, it's a little bit more sort of collective, with lots of cells having lots of neighbors, whereas here it looks much more sort of single file, similar to one of those other movies that I showed you earlier. The kind of false color scale that you can see in the top here is basically an indication of the number of neighbors that the cells have. And also in the spheroid context, we can see some diversity of pattern. So we can then start to try to quantify this pattern in terms of neighbor numbers, invasive extent, whether the kind of strands are tapering or have a uniform width, as it were, whether they get fractured. And using all these sort of metrics, we can then look at it in sort of multi-dimensional space. And in the bottom, you can see kind of two PCA plots, just looking at the diversity of invasion patterns that we can see. And just to sort of highlight so this number 1 here corresponds to this sort of dot here, and this is number 2, which corresponds to this, and so on and so forth. So I think this sort of tells us that we can get diversity in the model. It also, because these dots are kind of relatively kind of contiguous, again suggests that there's a kind of continuum of different ways that these cohorts of cells can invade, not kind of discrete categories. But with this in mind, we can then start to look a little bit deeper and really unpick which are the parameters that are driving these different types of behavior. So I'll show you a few sort of plots like this. And now what we've done is taken those PCA plots and then we just overlay the kind of the strength of the input parameter for a particular feature in this kind of in this case false sort of science scale. So in this case, sign is representing the extent of proteolysis that the cancer cells can do. And what you can see is that there seems to be a sort of broad association between the high protease region and the sort of dimensions in the PCA plot that are associated particularly with neighbor number and to a slightly lesser degree invasion score. But interestingly, the maximum depth, which is kind of the overall distance that the cells are able to go, didn't seem to be terribly well correlated with the amount of proteolysis that the cancer cells were able to do. And the plot on the right just shows you the same thing for spheroids. If we look at this in a sort of slightly simpler way, we can plot, one variable against the other. So you can just see that this kind of, feature of neighbor number seems to increase when we increase the proteolysis term, both in the spheroid context and in the organotypic. Whereas the invasion depth doesn't change that much. It varies a little bit in the organotypic context, and it even seems to drop a little bit in the context of the spheroid. So then we can return to our experimental system, take these cancer cells and then either CRISPR out MMP14 or we can overexpress it. So the KO is the knockout and the OE is the overexpression. And then we can measure various of these kind of strand characteristics. And what I hope you can appreciate and in line with the model prediction is that MMP14 leads to slightly thicker strands that you can see kind of indicated here compared to the dimension of the wild type strands. And if we look in the MMP14 knockout, if anything, they're slightly thinner. So this is kind of plotted here where we're looking at the strand width and it increases as a function of MMP activity. And the same thing holds true, albeit with a slightly shallower slope in the spheroid context. Intriguingly and kind of actually consistent with what the model was telling us about the spheroids is that actually overexpressing the MMP actually doesn't increase the strand length or the kind of maximum extent of invasion, actually because you're getting basically a broader kind of pushing front, so that the cells are kind of going more side by side and actually the greatest extent of distance isn't increasing. So this is, I think, an interesting, and somewhat counterintuitive prediction that the model made that we were able to validate experimentally. We've also obviously had a look at the relationship between the cancer cell matrix adhesion and pattern formation. This is actually a little bit less clear in terms of, you can see here, particularly in the organotypic, that the bright green dots are kind of distributed almost all over the PCA plot, which tells us that things are not so clear. And when we've tried to experimentally perturb this by CRISPR deleting integrin beta-1, we haven't seen such strong kind of effects. What we do also need to do is we need to eliminate integrin beta-3, and this is work that is kind of currently ongoing, but I won't kind of dwell on this any longer. Suffice to say that actually the adhesive properties of the cancer cells to the extracellular matrix has a complex and maybe not such a strong relationship with the pattern as we might have expected. So then the kind of third major parameter that I'm going to tell you a little bit about is that of the cancer cell adhesion to each other. So I should have sort of said at the beginning that we're working with squamous cell carcinoma, and these is a type of cancer that really has clear retention of E-cadherin in the great majority of cases and retains conventional adherence junctions, albeit they may be a little bit disorganized. So what I hope you can kind of see here is, again, now the cell-cell adhesion parameter is shown in magenta. These dots kind of cluster over to the right as we're looking at it here. That it does seem to be associated with invasive extent, suggesting that there's a positive relationship between the formation of these cell-cell junctions and the ability of these cells to invade. Certainly in the organotypic context, the kind of thing that is scoring more strongly in the spheroid context seems to be related to the invasion score, not actually the maximum depth, and I'll return to this in a couple of slides' time. So the kind of major kind of take home from these sort of predictions is that cell-cell adhesion seems to favor invasion, but there does seem to be some context and some difference between the organotypic and the spheroid assay. So just to kind of simplify this sort of prediction a bit, where we're looking at the kind of the strand length, which is the extent of invasion in the organotypic, the prediction is that there will be a sort of positive association with high SCC invasion, giving high cancer cell adhesion, giving you more invasion, whereas it will be relatively flat in the context of the spheroid invasion assay. So again, we've tested this. We've done this in a couple of different ways. I'm just going to show you the data where we've CRISPR'd out alpha-catenin to disrupt the formation of cell-cell junctions. And this leads to a very significant reduction that I hope you can see here in the invasion in the organotypic context, but actually in the context of the spheroid assay, there isn't really that much of a difference in strand length. So this is supporting, again, what the modeling was telling us. So this kind of intrigued us, and we wanted to know why this might be the case. And one thing that we kind of thought about when we were going through this work is that actually the way the model is configured and also the way the experiments are configured is that in the organotypic context, you have all the matrix down here, and also all the serum is down here. So all the kind of chemotactic goodies are in this direction. So when we built the model, we put sort of a chemotactic queue kind of at the bottom, as it were. Whereas in the spheroids, where the spheroids are kind of encased and sort of surrounded by both the serum and the extracellular matrix, we put a radial kind of queue, in terms of the directionality. And this kind of leads to a somewhat obvious thing, that if you've got cell-cell adhesion and you've got, I'm just going to draw two cells for the sake of simplicity, but they're trying to go in different directions, but they're stuck to one another, the net result is that they're not going to go very far, suggesting this is the source of the a potential negative relationship between cell-cell adhesion and invasion. But what happens if we just kind of conceptually take this assay and we make the directional cues uniform? So now the prediction is that actually the positive relationship between cancer cell adhesion and strand length will reappear similar to in the organotypic assay. So this really kind of suggested to us an idea that perhaps what the adherence junctions are doing is actually favoring favoring invasion when there is a uniform kind of directional cue present, so that actually these adherence junctions might be promoting coordination between the cells. And this kind of took us back to some work that we'd done almost 10 years ago, led by Cristina Hidalgo, who'd been able to kind of identify that these kind of small groups of the cells have this kind of supracellular organization of actomyosin around the outside of the group. And this is, as you might not be surprised to learn, dependent on the presence of adherence junctions. So this kind of supracellular coordination of the actomyosin cytoskeleton is disrupted when we disrupt adherence junctions. So what we wanted to do was kind of basically ask, is there an orthogonal, a different way that we could achieve the same thing? And actually we exploited a tool that was actually developed by Mike Olson a few years ago, which is basically a constituent or an inducible kind of form of ROC, the kinase that drives actomyosin contractility, because actually you get ROC suppression at the interface between the cancer cells in the wild type context. And what we were able to show is actually that by disrupting this kind of supracellular actomyosin contractility or cytoskeleton, we had a similar reduction in strand width as when we depleted the adherence junctions components, suggesting to us that alpha-catenin and adherence junctions more generally promote the kind of widening of these invasive strands by enabling the coordination between the cells. And this is why they're actually kind of pro-invasive when there is a uniform directional cue present. Just in the last sort of 5 or so minutes, I'd like to kind of transition a bit to kind of considering actually the intersectionality between these different variables and how there might actually be a link between strand width and strand widening and actually the propensity of these carcinoma cells to grow. So first up, the kind of relationship about what happens if we play around with multiple parameters at the same time. So here I've already shown you that if we overexpress MMP14, we get these wider strands that you can see here, and this is kind of quantified here. It turns out that that strand widening kind of along the lines that I've already been telling you, depends upon the adherence junctions. So for proteolysis to generate a wide strand, you need those cell-cell junctions to be present, because you get much narrower strands, as you can see, if you CRISPR out alpha-catenin at the same time as overexpressing MMP14, and this is kind of shown here. And then, the final thing that actually was a little bit of a surprise to us, but actually might provide some of the explanation as to why it seems important for these SECs to retain their cell-cell junctions, is that there turned out to be a relationship, particularly when there were no fibroblasts present, between some of these parameters and the ability of the cancer cells to grow. So I haven't really told you about growth at all so far in the talk, but obviously, as we're modeling cancer, these cells have a propensity to grow. And in line with all the experiments we've done for the last 15 years or so, whenever we play around with the system in the presence of CAFs, there's very little relationship between the growth of the system and any of the sort of things that we've played around with when we have CAFs present. Intriguingly, the model kind of predicted to us that actually this matrix degradation term by the cancer cells would be very important in the context of the spheroid assay for growth, but not in the organotypic. And this is, I think, not too hard to understand, because the spheroid is kind of encased by extracellular matrix, whereas the organotypic has free space above it where the cancer cells are able to grow. So this kind of made us think that actually there might be a relationship between strand widening, which obviously generates space in one direction, and the ability of these cells to grow. So actually we tested this just by putting these cells in three-dimensional collagen, now in the absence of fibroblasts, so you don't get too much invasion. They stay as relatively round balls. But what I hope you can appreciate is that actually, all the same manipulations that make the strands narrower reduce growth, and all the same manipulations that make the strands wider actually boost the growth, in particular, the overexpression of MMP14. And this is just quantified down here. So you're looking at the green one, which is the MMP14 overexpression. And once again, this depends on the presence of cell-cell junctions, because if you deplete them, which is this one here, you see a great reduction in growth. In the kind of very final bit of data I'd like to share with you, we've now also grafted these same cancer cells into actually the dermal space in the ears of mice. This seems to be a relatively sort of spatially restricted environment, as one might imagine, given how thin the ears are. And it also seems, given what we're seeing in terms of the growth dynamics, which is a very nice recapitulation of what we were seeing in vitro and some of the features that the model was predicting, is that it seems to be somewhat limiting in terms of the ability of the fibroblast to generate space, and it's very much dependent on the protease function of those cancer cells, but also dependent on their ability to generate or to form adherence junctions. So this is looking at growth in the intradermal space where they were injected, and this is now also looking at the ability of those cells to disseminate and grow in local lymph nodes. And once again, we see that MMP14 is kind of key to this, but ultimately it depends on the ability of the cells to make adherence junctions and to generate that space. So I'll just kind of conclude with some take-home messages, and then I'll be very happy to answer questions. So I hope I've been able to kind of convince you that multiple different collective invasion patterns can be present in the same tumor. We don't see any evidence for discrete or distinctive collective invasion categories. It seems to be a continuum. In terms of what are the parameters that determine whether the cells kind of invade collectively, as a wide kind of pushing strand or as a very thin strand, matrix proteolysis seems to be key in determining the strand width, although intriguingly not the extent of invasion. It seems clear that adherence junctions in the cancer cells enhance invasion, but only if the directional cue is uniform. And it seems that this is related to the ability of adherence junctions to lead to supracellular coordination of the actomyosin cytoskeleton. I've also kind of told you that the proteolysis-driven strand widening requires adherence junctions, and kind of just sort of to finish with, a couple of, I think, unexpected or at least partly unexpected observations. When we were doing all these manipulations, actually the thing that makes the cells most kind of mesenchymal in the EMT kind of way of thinking about things is giving them high proteolysis, but no kind of cell-cell junctions. And actually it turns out that in a range of different kind of contexts, these cells are really not the most invasive. And even in the context of growth, they're not even the most able to grow. We think because they have some defects in being able to generate sufficient space for growth. And this is, kind of bringing me to my final kind of point that I'd like to reiterate, that there does seem to be a sort of mechanistic connection between the tract widening and actually the ability of these tumors to grow effectively. And this might be the reason why actually this type of squamous cell carcinoma typically retains cell-cell junctions, even as it metastasizes through the lymph nodes and to other sites in the body. So I will finish there by thanking all the people who've done the work. The computational modeling was really led by Rob Jenkins, and all the experimental work was done by Takuya Kato, who's now back in Japan. We've had great help from the other people listed, and I'd really like to thank both Selvam and Kevin as clinical collaborators who've given us access to the patient tissue, which really has been invaluable. So thank you, and I'm happy to answer questions.
Thank you very much, Erik.
I guess I should stop sharing my screen. Is that right?
It's up to you. Yeah, you can stop sharing. That's fine. Great. So we've got a load of questions, so we're going to try and get through as many as we can. We'll start with a question from Fatima on YouTube, who's asking, is pulling of the matrix by CAFs also taken into consideration when evaluating migration of cancer cells?
So that is a great question, and that is exactly what Hamid is talking about very shortly. So I will just take it as a setup for him. But yes, it happens, and yes, it's important.
Great, thanks. We have a question from Yi Lim Lee regarding computational shape and direction of the invasion to explain the increasing trend in the SCC adhesion. It seems like the computational model of organoids is more circular than a spheroid, but the direction of the invasion shows that the organoid actually migrates in one direction while the spheroid is migrating in all directions. Could you explain why the assumption of computational shape, how circular they are not associated with the direction of the migration?
Sorry, I'm just trying to digest the question.
I can jump in here.
Yeah, thanks.
You remember the graph on, I think it was like towards the end, where you showed there's increasing trend on the SCC interaction for the, that it was because you're assuming they're migrating out in one direction, while spheroid, they're more linear because they're migrating in all directions. But in your computation model, it looks like your organoid is more circular shaped than the spheroid. So
yeah. So I think one thing I perhaps didn't make clear was the images that I'm showing you are effectively a 2D cross section of the 3D model. So the model is three-dimensional, but the images that are shown are just a 2D section for presentation.
So you're assuming both of them are in perfect circular shape, or they're somewhat similar in circularity?
Yeah, so in both cases, and I hope I'm answering the question, the kind of the ball of cells gets initiated as effectively, a spherical kind of object. Then, due to some stochasticity in the model, it doesn't necessarily, retain its perfect sort of spherical shape, but it can sort of form some, sort of bulges. Particularly if you have fibroblasts present, they tend to be kind of instructive in the formation of those bulges.
Sounds good. Thank you.
Okay.
A couple of questions from Rita Brata. The first one is asking if the model is able to predict when there are no CAFs, but there are chemical cues.
Yeah, so good question. One thing we've done, I didn't have time to go into the details, is the CAFs kind of make sort of tracks. This was, work we did several years ago. We've also kind of run the model where it's kind of got like a tiny, thin little track, but no CAFs, because the CAFs kind of make it a bit noisy because they generate some stochastic behavior. And broadly speaking, we get the same results if we use that context. And actually, they look a little bit cleaner than if we have the CAFs present. So yes, that does hold up.
And the second question was saying that in physiological settings, CAFs would infiltrate the tumor. And they're asking if you have a system where you have CAFs within the tumor itself and what you would expect to see.
Yeah, so in the organotypic system, the fibroblasts are initiated in the matrix underlying the layer of cells. In the spheroids, the two cell types are initiated in the same place together. So they kind of recapitulate two slightly different contexts, one where the two cell types are separate and the other where they're mixed together. So I think we do have that system. That's how the spheroids are kind of initiated.
Great, thanks. From Bob Fisher, in the context of understanding actual metastasis, does it make more sense to ask what the outlier cells / group of cells, the most migratory ones, do, rather than use the model to interrogate the mean or average behavior, particularly with respect to the adhesion behavior and proteolysis?
That's a great question. We've done a little bit of looking at that. What we haven't done, what we played around with, but we haven't kind of got to the bottom, is actually modeling where you have cancer cells that have variability in those parameters, within the population. I think that's probably the most interesting way to go. But it's a great question. I completely get where it's coming from. We should look in more detail at it, I think is the simple answer.
We'll do a couple more questions and then we'll go to Hamid because we've got so many questions. Maybe we can come back at the end. So Marco is asking if the model is able to predict changes in CAF that can drive different migration modes of cancer cells.
Hi, Marco. Good question. So we also played around with three CAF parameters, I think. One was CAF matrix adhesion. So if the CAFs are better able to interact with the matrix, generally you get more invasion. Didn't seem to do too much to the pattern. We played around with CAF cancer cell adhesion as a parameter, kind of following on the work that we did with Xavi. That kind of scores as important if the fibroblasts are good at interacting with the matrix and the cancer cells have strong cell-cell adhesions. Effectively, it then sort of makes a glue between the fibroblast that's moving away and the cancer cell so that it can drag them behind them. But we didn't see really big kind of effects of CAF parameters on pattern, so to speak. It was more an extent of invasion.
From Giorgio Sita, the model seems ideal also to interrogate the impact of heterogeneity. Have you done this in any way?
Yeah, so we have done that, and we kind of get interesting things around kind of these different cells sorting into different populations, kind of going back to literature sort of 30, 40 years back. So if you mix the cells that make adherence junctions with ones that don't, they kind of self-sort. We've got some intriguing data that suggests that cells with low protease activity might be able to non-cell autonomously interfere with the invasion of cells with high protease activity. That's kind of intriguing. Almost they kind of form a sort of little barrier around the cells with high protease activity. So great question. We played around with it a bit. There's interesting stuff, but we haven't, it's not quite sort of prime time, as it were. I should probably hang around at the end if answer more questions, because I'm aware of time and Hamid deserves his moment.
Yeah, absolutely. So thank you very much, Erik.
But I